Today we discuss the second unit from Plus 2 Biology, Genetics and Evolution. This unit has 3 chapters: Principles of Inheritance, Molecular Basis of Inheritance and Evolution. Altogether, these 3 chapters give about 18 marks question for your board examination. So consider this as an important unit. And the first chapter, Principles of Inheritance and Variation. So these are chapters you feel very interesting. Let us move on to the first chapter, Principles of Inheritance and Variation. So you have already learned what is genetics. Okay, so tell me, what is genetics? Yeah, genetics is the study of heredity and variation. So genetics is a branch of biology which deals with heredity and variation. So in order to understand genetics, you should know what is heredity and what is variation. So heredity means what? You may be better familiar another term, inheritance. Okay, so this is a commonly used word, inheritance. So inheritance is the basis of heredity. So heredity refers to transfer of characters, the process by which characters are transferred from parents to progeny. Okay, so inheritance is a process, you know that, you are familiar. And you know a human gives birth to a human baby. A dog gives birth to a pup, never a calf or a kid. Similarly, a mango seed will be forming a mango tree. Okay, mango plant only it produces. So this resemblance is coming under heredity. How progeny resemble their parents is discussed under heredity. So heredity refers to the resemblance. Okay, like to be, yet like. Okay, so we can say inheritance or heredity, it deals with how characters are transferred from parents to progeny. Okay, so how you define heredity? It's a process of transfer of characters from parents to progeny. Is it okay? It's a process of transfer of characters from parents to progeny. So how these characters are passed? You know this already. That's why I asked. Okay, so how are these characters passed from parents to progeny? You have some idea. You know the units of inheritance are genes. Okay, so what are the units which transfer these characters? Are genes. So genes are the carriers. Genes transfer our characters to our progeny. Okay, so what does heredity means? Okay, it deals with inheritance, the transfer of characters from parents to progeny. It is accomplished through genes. Okay, then let us start with the next term, variation. What is meant by variation? So here we discuss about the resemblance. Oh, so the other part is variation. We are not exactly similar to our parents. We are not the ditto of our parents. We have so many differences. Okay, how we differ from our parents is discussed under variation. Okay, why are the progenies exactly similar to their parents? That is discussed under variation. But tell me, why are the progenies exactly similar to their parents? Is there any option for the progenies to resemble their parents exactly? Yeah, you have already learned asexual reproduction will give clones. Okay, morphologically and genetically identical offsprings which resemble their parents. Whereas, you know, here the differences come. Okay, from sexual reproduction. Okay, even our foreparents, our ancestors who lived in about 8000 to 1000 BC understood this fact. They even understood that the variation is hidden in sexual reproduction. By the time they were doing agriculture and animal husbandry, they understood that while the progeny differ from the parents is due to sexual reproduction. Something in sexual reproduction make the progeny differ from their parents. Tell me what? You have already studied the chapter meiosis and you know. Okay, you know during sexual reproduction, both the characters, maternal and paternal characters are mixed to form the progeny. Okay, so that there is difference, okay, automatically. Anyway, sexual reproduction brings variation mainly through the meiosis division. Okay, that the division meiosis is occurring only at the time of sexual reproduction during the formation of gametes. You know that. So, in meiosis, during prophase 1, there is a stage called Pachytene. Okay, Pachytene stage, and during all things you have learnt in +1. And in Pachytene stage, there occur crossing over. So, keep in mind, crossing over is the process which is responsible for variation. Okay, the main process. There are other processes like mutation, okay, gamete fusion, so many things. Okay, one of the major causes of variation is crossing over. Then tell me what is crossing over. That also everyone knows because you have already studied it. You have studied what about the homologous chromosomes and its crossing over. You know, homologous chromosomes are the chromosomes which are exactly similar in size, shape, everything. Okay, then, so these two are homologous chromosomes. Okay, a pair of homologous chromosomes. Then what is meant by crossing over? Then how crossing over bring this change? Tell me. Okay, when these homologous chromosomes exchange their segments, I mean this segment is exchanged to here and this one is exchanged to here. When the homologous chromosomes exchange their segments, that process is called crossing over. Exchange of segments between homologous chromosomes is called what? Crossing over. Then you can very easily understand the genes will be shuffled and this is bringing variation. So one of the major reasons for variation is what? Crossing over, which takes place during gamete formation or during meiosis division. Okay, so it takes place during Pachytene stage of meiosis. It's very easy idea because you have already studied. And not only this, that you know mutation occurs, that may be a reason for variation. This all finally lead to evolution. Okay, all these things will be clear by these 3 chapters. After completing this unit, you will be very much thorough about the genes, evolution, everything. Okay, and this is just an introduction section only, and we will move on to the Mendelian genetics. Before that, just understand genetics is a branch of biology which deals with heredity and variation. Heredity refers to how we resemble our parents, and it is especially through genes. It is achieved through genes. These are acting as the units. Okay, then variation refers to how we differ from our parents, and it is mainly due to the crossing over which takes place during gamete formation. Okay, and also gamete fusion and mutation can be other reasons for variation. And finally, so variations are leading to gradual evolution. Okay, then we can just discuss genetics under 3 areas. Genetics is divided to 3 parts: pre-Mendelian, Mendelian and post-Mendelian genetics. And as per your NCERT textbook, there is not much concern of pre-Mendelian genetics given. Okay, so we start with Mendelian genetics. The pre-Mendelian genetics deals with some of the discoveries by scientists before Mendel. Anyway, we start with Mendelian genetics. Okay, so when we discuss Mendelian genetics, you have already learned in 9th standard about Gregor Johann Mendel. Okay, so who is Gregor Johann Mendel? Gregor Johann Mendel is known as father of genetics. Okay, you know, he was not a great scientist. He was a monk. Okay, was born in 1822 in Austria, the present Czechoslovakia, in a small village called Heisendorf, and he was a monk in Brand City, one monastery of Brand City. Then actually he was not a very great scientist, but he could do something, a pioneer attempt to genetics and develop a new field of biology, genetics. Then he has conducted his experiments in simple garden pea. Okay, the experimental material of Mendel, Gregor Johann Mendel, father of genetics, was garden pea. What is the scientific name of garden pea? It is Pisum sativum. This was a board exam question. Okay, the scientific name of garden pea is Pisum sativum. The P should be in capital letter, S should be in small letter, and should be separately underlined. You know the rule. Anyway, listen to me. When Mendel conducted his experiments using garden pea, later when other scientists evaluate Mendel, the major reasons for his success, they evaluate as. One by one we discuss what are the major reasons for Mendel's success. Okay, the first one we say Mendel conducted long-term experiments. He didn't stop his experiments within, okay, one or two experiments in short term. He has conducted long-term experiments for about 7 years. Okay, in order to produce pure lines, itself, he took 2 years. You will understand what are pure lines. Anyway, the first point of success of Mendel is he has conducted long-term experiments. Took about 7 years for his experiments. And second thing, you will clearly understand what are pure lines. Mendel conducted all his experiments using pure lines. And for that production itself, he took about 2 years. What do you mean by pure lines? When Mendel selected any plant character or a trait, tall or dwarf, he has ensured that that character is pure. So, for example, when he selected tall plant for crossing, he made it pure tall by continuous self-pollination for many generations, about 2 years. Similarly, he took dwarf plant, or violet flower, white flower, every plant he self-pollinated for many generations to produce pure lines. He was sure that there is no any character, tall character is not mixed up with dwarf. There was no any natural pollination. He self-pollinated the plant and made sure that this is a pure line. And you will have some more better understanding of pure lines and segregation later. Now just understand, it was a long-term experiment he did, and in order to produce pure lines, he took about 2 years, and he conducted all his experiments with the pure lines. And another thing, he has used, always for his experiments, he has used opposite traits or contrasting traits. If, for example, if one is tall, he has taken the other one as extreme dark. Okay, likewise, always contrasting features he selected for his experiment. That is another reason for his success. And also, he has selected large sampling size for his experiment. The fourth reason for Mendel's success is he has selected large sampling size. He didn't limit his experiment with 1 or 2 plants. His sampling size was large. Okay, and you know Mendel was the first man who introduced mathematical logic and statistical analysis to biology. These all are the reasons for Mendel's success, and he has got a good experimental material, Pisum sativum. This is also one of the reasons for Mendel's success. Anyway, let us discuss one by one what are the major causes or reasons of Mendel's success evaluated by other scientists. Okay, he conducted long-term experiments and he has already produced pure lines and conducted experiments with pure lines, and he always used plants with contrasting traits. and he has selected large sampling size for his experiment, and he was the first man who introduced mathematical logic and statistical analysis to the biology, and he used Pisum sativum as a material, and this material was very easy to, the plant was easy to handle, and the plant was easy, its flowers were conspicuous to carry out any pollination, okay, and it is a self-pollinating one, okay, and it's a bisexual plant, then only it do the self-pollination, okay, a bisexual plant, self-pollinating one, and it completed the life cycle quick, okay, and it provided number of traits with contrasting, okay, features. He could develop, or he could find many different varieties of this plant. These all are the reasons for Mendel's success. Okay, and I think we need not stress it again. We have again so many important topics to discuss. Okay, so about the Mendelian genetics, what is Mendel's material, what made Mendel a successful man in his experiment. So all things are clear, I think. Then let's move on to Mendel's work. Mendel started his experiment in 1856. He was born in 1822. Okay. Then he started his experiments in 1856, and about 7 years he took to publish his works. Okay. Anyway, he belonged to Austria, present Czechoslovakia, and he has always selected many contrasting traits for his studies. That is considered as one of the main reasons for his success. Anyway, which are the 7 contrasting? Anyway, altogether Mendel has found about 34 different traits in Pisum sativum, and he used about 22 of them in his studies. Okay, among that, the 14 main traits we will discuss here. Okay, 14 main traits of Mendel. So, Mendel has studied the first character, stem height. Okay, stem height is the character. Okay, it has one character, its expression we can call as trait. Expression of a character we can call as traits. Okay, the character is stem height. It is expressed as tall and dwarf. The two contrasting characters Mendel took are tall and dwarf. The stem height is the character. Two traits are tall and dwarf. The second character, flower color. Flower color is the second character. Violet and white. Violet and white. Third character, flower position. Flower position, axial and terminal. Axial and terminal are the flower position he has studied. Then fourth one, pod. Pod shape. Pod shape he has noted, inflated and constricted. Pod shape, inflated and constricted. Then fifth one, pod color. Green and yellow are the pod color he has taken. Green and yellow. Sixth one, seed shape. Seed shape. Round and wrinkled. The two opposing traits he has studied. Round and wrinkled. And seventh one, seed color. Yellow and green. Okay, some more things to remember, study here. See, Mendel, for his main experiments, he had used these 7 characters with these contrasting traits of them. 7 characters, 14 contrasting traits. Stem height, the 2 contrasting traits, tall and dwarf. Then flower color, violet, white. Flower position, axial, terminal. And the axial position, flower position means in the axial the flower is, that's a common type. Then the terminal one, axial, terminal, flower position. Then pod shape, inflated, constricted. Inflated pod shape means the smooth with a smooth surface. Constricted pod shape means like this. Okay, pod shape can be inflated or constricted. Then pod color green, yellow. Then seed shape round seed or wrinkled seed. Okay, round or wrinkled. Then seed color yellow or green. Then this all when you learn, just understand all the first written. Tall is dominant over dwarf. What is dominant recessive you will understand. I mean the major one is tall, dwarf is recessive. Similarly all first written are dominant traits. We will discuss it. Okay, so the 7 contrasting characters, I mean the 7 pairs of contrasting traits studied by Mendel are this. Easy I think. Now I need to stress it again I think. Okay. Anyway I give the notes too and you will understand it better. Okay. So Mendel has studied all these features in garden pea and he has conducted so many experiments all with plants of contrasting traits. Among that the 14 major contrasting traits he has studied is discussed here. Okay, then let us move to the major portion of Mendelian genetics. Mendel has conducted monohybrid crosses, dihybrid crosses, trihybrid crosses, etc. And here first we discuss a monohybrid cross conducted by Mendel. Monohybrid cross. So you have learned it already. Still, can you say what is a monohybrid cross? You might have studied. It's a cross in which only one character is taken into account. Okay, it's okay. When the breeder is studying the inheritance of single character at a time, that cross is called the monohybrid cross. For example, the plant may be having number of other features, but the breeder, the experimenter is focusing on inheritance of only one character. Stem height only he is considered. I mean, tall dwarf only he is considered. Maybe what is violet flower, seed different, nothing into account. If the plant breeder consider, okay, if the scientist consider only one character at a time, that cross is called a monohybrid cross. So, a monohybrid cross is a study of inheritance of single gene. Okay, one gene inheritance is studied here. That is why we say only one character is focused at a time. The first monohybrid cross by Mendel, Mendel crossed tall pea plant with dwarf pea plant. Mendel crossed a tall pea plant with a dwarf pea plant and he produced a generation and he has named this generation as first filial generation. Okay, this were the parental generation. Mendel crossed a tall plant with a dwarf plant and produced first filial generation. He called it as F1. Okay, and he found only tall plants in the F1 generation. All the plants of F1 generation were tall. And secondly, he has self-pollinated F1 tall plants, selfing. Okay, that means its own pollen grains are deposited to the stigma. Selfing is done in the F1 plant and he produced the second generation named as second filial generation, F2 generation. Second filial generation. Interestingly, he found that about 75% of them, F2 generation, were tall plants, whereas 25% of them were dwarf plants. Okay, that means in his monohybrid cross, in the first generation, when he crossed two plants of opposite traits, tall and dwarf, in the first generation, all the progeny resembled one of the parents only, whereas the other parental character never appeared. Whereas when he raised the second generation by selfing of the F1, self-pollinating the F1 generation, he could produce both the characters. Okay, about 3/4 of his first experiment. In the F2 generation, 3/4 of them were tall and 1/4 of them were dwarf. Okay, and in order to explain the result as a conclusion, Mendel has given so many concepts. Okay, according to Mendel, one character is controlled by one gene. He didn't use the term gene, he has used the term factor. According to Mendel, one character is controlled by one factor. Okay, and he says the factor exists in pairs. Okay, factor exists in pairs. Now we know that this is gene and this is a pair, allelic pair. Okay, so in order to explain his result, Mendel has introduced his concept of factors, and factors are present day genes and factors pair. Members of the factors now we call alleles, and we can use the similar term to study that. And anyway, we are just using some symbols based on Mendel's idea. Mendel used the same symbols for his studies, and so we drew the Mendelian monohybrid cross in a new way. Okay, using symbols. The parental generation, okay, we represent using symbols. Okay, first plant, tall plant. Okay, the second plant, dwarf plant. One of this is male, the other one is female. This one we can take as male plant, this one as female plant. And when we use symbols, one character, for one character there are two members of the factors, two member factors according to Mendel. So we can say there are tall character, there are two alleles. So this is one gene, it has two alleles. Why we write capital T here? Because Mendel has produced all pure lines for his experiment. He was sure that this character is pure for tall. So he has used the capital letter T for tall and small letter t for dwarf. Since they are pure, he has used the term similar terms. We will discuss it, homozygous, heterozygous cases. Then according to Mendel, when these plants produce the gametes, when they form gametes, gametes receive only one of the members. Out of the two alleles, only one is gone to gametes. In order to explain this, he has later produced law of segregation. We will discuss that law later. Anyway, these are the parents. Tall parent is represented by capital T. Dwarf parent is represented by small t. And when they form gametes, gametes carry only one of the two alleles. That means this pair is segregated, separated in gametes. Gametes, you know, gametes are having haploid number. So out of the total chromosomes, only 50% will be in the gametes. So there is a segregation. Okay, the gametes are represented like this. And when we do the gametes combination in a Punnett square, I mean the square board used to work out this gamete combination was developed by Reginald C. Punnett. Reginald C. Punnett. So this square is known as Punnett square. Punnett square. Okay, in your textbook the square is drawn this way. Okay, just put that. So anyway, when we use the Punnett square for working out the gamete fusion, we write the male and female gametes outside the margin. That means you can do the female gametes here, male gametes here. The male gametes are capital T, capital T. Gametes are always written inside a circle. Then the female gametes are small t, small t. Only this character we consider. All other character gene it will be having. Okay, so anyway, when we write the combinations inside, this is, I mean, this male gamete with this female gamete combination, capital T, small t. This male gamete with this female gamete combination, capital T, small t. Here, this male gamete, this female gamete, capital T, small t. And this male gamete and, I mean, leftmost and rightmost, that is small t. I mean, this capital T, this small t. And these are the combinations, how to write the combinations. And all these plants were observed as tall. Okay, all of them were tall. And here comes the second theory, we will discuss it. Tall. All the plants of F1 generation were tall. So Mendel could observe all of the F1 plants were tall. Okay, so the result we write all of them tall in the F1. Okay, then this is about the Mendel's first step. That means he crossed a tall plant with a dwarf plant and produced F1 generation and found all of them were tall. And how to raise the second generation? In order to raise the second generation, one of this plant he has taken and he has self-pollinated it. Okay, this is a tall plant and this has crossed with the same tall plant. That means self-pollinated. Its own pollen grains are deposited on the stigma. We can write the step as F1 selfing. All the steps you have to write, that is must. Parental, gametes, F1 generation, F1 selfing. During F1 selfing, when this one is male, other one is female. When there is gamete formation, you know what happened? The allele pair separate. There are these two gametes. I mean the gametes carry capital T, small t. Here also the gametes carry capital T, small t. This similar gametes they produce. Okay. Then the next one we should show the cross in a Punnett square. I'll just show the Punnett square here. Okay. The F2 generation Punnett square. Okay. Then when we draw the Punnett square, we can just do the gametes outside in a circle. Okay, these are the female gametes or female or male, okay, whatever. You can just, what is given here, the same way you give it. Then this is F2 generation. It should be written to the left side. Okay, like in the same line you write parental gametes, F1 generation, F1 selfing, F2 generation. And in the F2 generation, when we get the combinations, we get first one male gamete, capital T, female gamete. Okay, capital T is the combination. Second one, capital T, small t is the combination. Third one, capital T, small t is the combination. The last one, small t is the combination. Okay, then let us discuss what are the appearance. This is appearing as tall. This one also as tall. This is as tall. We will discuss how it is. Okay, then when Mendel observed the F1, all of them were tall, whereas in the F2 he found tall and dwarf. Okay, in the ratio 3:1. I mean 75% of the plants were tall and 25% were dwarf. In the 3:1 ratio, he obtained the tall and dwarf in the F2 generation. And this is how the Mendelian monohybrid process is working out. And we will explain the theory behind it, the principles proposed by Mendel. Anyway, just see that when you work out monohybrid process, you have to keep a left line writing parental, gametes, F1 generation, F1 selfing, and then F2 generation. Okay, and in the here middle section you have to write the first one parental generation showing cross and its gamete splitting of the gametes. Okay, then the Punnett square work, then its result should be written separately. Then the second Punnett square work, its result should be written separately. Okay, then there are so many things to understand from this about the phenotype, genotype, dominant, recessive, homozygous, heterozygous, so many terms to understand from this. I am just removing this part that I will keep to just to understand the concepts. Okay, just rubbing this part. So this is the Mendelian first monohybrid cross, tall versus dwarf. All the F1 were tall, whereas in the F2 the dwarf character also appeared. And in order to explain the reason, he proposed the law of dominance, law of segregation. And the Mendel is very famous for principles of inheritance. And among these principles of inheritance, the first two laws, law of dominance and law of segregation, he has introduced immediately after monohybrid process. It will be clear from the monohybrid process. The third law, law of independent assortment, is meant for dihybrid process, that we will discuss later. Anyway, we are moving to principles of inheritance by Mendel. The first law is law of dominance. Mendel's law of dominance. Okay. According to Mendel, there are three main postulates for this law. Okay, the law of dominance. According to Mendel, characters are controlled by discrete units called factors. Characters are controlled by, now present by genes. Okay. According to Mendel, characters are controlled by factors. Okay, that's the first proposition. Second one, factors exist in pairs. Mendelian factors exist in pairs. You know now they are allelic pair. We will discuss gene alleles or concept we discuss. Then the third postulate is, in a pair of dissimilar factors, if a factor has a dissimilar pair of members, one member dominates the other. Okay, in a pair of dissimilar factors, one member dominates the other. That only expressed, the other one is getting suppressed. Okay, one member dominates the other member. The member which is dominated is called dominant, whereas the member which is suppressed is called recessive. We will discuss it with the same example, this example. Okay, so according to Mendel, when we take the character, when we use the symbol capital T and the small t for stem height, okay, these are the factors. Okay, different combination of the factors. It can be either capital T combination, or small t combination, or capital T small t combination. These three combinations only possible. Okay, and if the alleles are same, if the factor members are same, such a combination is called homozygous. Okay, if the allele combination, capital T and capital T, small t and small t, if the alleles of a gene or a factor pair are same, then that is called homozygous condition. Then this condition is called, a pair of dissimilar allele cases is called heterozygous condition. I think these two terms are very clear. It's very important. In homozygous combination, the alleles are same, whereas in heterozygous combination, the alleles are dissimilar. Okay, and in this particular example, capital T, small t combination come homozygous, capital T small t combination comes heterozygous. And in this dissimilar combination, that means in a pair of dissimilar members, dissimilar alleles or dissimilar factor members, one is getting expressed and the other is getting suppressed. Only one of the two alleles, when a dissimilar pair come, when two alleles come together, dissimilar alleles come together, only one member is expressed. That member is known as dominant. Okay, whereas a suppressed one is called recessive. Okay, so make it clear, in a pair of dissimilar factors, only one factor member. Now we can call one allele gets expressed and the other one is suppressed. The allele which is getting expressed is called dominant. Okay, the allele which is getting suppressed in heterozygous condition is called recessive. Okay, for this are the four new terms studied: homozygous, heterozygous, dominant, recessive. Is that clear for you? Okay, regarding law of dominance, this dominant recessive ideas should be clear. And here also in a pair of dissimilar allele combination means heterozygous condition. Then only this concept will be clear. Law of dominance. Okay, according to Mendel's law of dominance, there are three postulates. Characters are controlled by discrete units called factors. Second postulate, factors exist as pairs. And third postulate, most important, in a pair of dissimilar factors, one member only is expressed. One member dominates the other. The first member is called a dominant, which is not getting expressed, is called a recessive. Okay? Then, one more thing to understand regarding, and these terms are familiar, I think, dominant, recessive, homozygous, heterozygous. And next class we will revise it once again, whether these terms are clear for you. Okay. Then anyway, see, you have seen tall with this condition, this is expressed as tall and this also expressed as tall. For dwarf, it is expressed, okay, it is small t, small t. Then, just listen, if a plant is there, tall plant is there, we are not sure whether it's this type of homozygous tall or heterozygous tall. Whereas if a dwarf plant is there, there is only one possibility, we are sure it is this combination. Anyway, this morphological expression is known as phenotype. How is the plant appeared is known as phenotype. Whereas this genetic makeup, the allele combination is called genotype. So you have studied two more new terms. The genetics is full with terminology. Okay, that I will tell you. Then see, tall characteristic is expressed by, it is expressed as tall, but we cannot say, it's genotype. Genotype is the hidden genetic makeup. Whereas how it is expressed is what? It's phenotype. About tall is phenotype, dwarf is phenotype. Whereas how is the genetic makeup is its genotype. Capital T genotype is expressed as tall. Oh, this is genotype, this is phenotype. So this is genotype, this is phenotype. So the tall characteristic has two genotypes possible. Dwarf has only one genotype possible. Phenotype is the morphological expression of a trait, whereas genotype is the inner genetic makeup, how the allele is, whether it is homozygous or heterozygous. Then here comes two types of ratios. That is what I have kept it as such here. When we write F2 ratio, you have to note down two ratios. Phenotypic ratio, genotypic ratio. Can you say now what is phenotypic ratio? Phenotype means the express feature, tall and dwarf. Okay, the same thing. Phenotypic ratio is 3 tall is to 1 dwarf. The phenotypic ratio of F2 is 3 is to 1. Okay, then what is genotypic ratio? Genotypic ratio, how many are capital T? Only 1. How many are capital T, small t? 2. And how many are this way? Small t, 1. That ratio is genotypic ratio. That means 1 capital T is to 2. How many capital T, small t? 2 capital T, small t is to 1 small t, small t. Genotype combination is a ratio. So the ratio is 1 is to 2 is to 1. The genotypic ratio of F2, monohybrid F2 is 1 is to 2 is to 1. That means 1 capital T genotype, 2 capital T small t genotype and 1 small t genotype. 1 is to 2 is to 1 is the genotypic ratio. I think it is clear. Phenotype refers to the morphological expression, expressed, how are the traits expressed. Genotype refers to the genetic makeup, how are the allele combination. Okay, whether it's homozygous or heterozygous. That is about the Mendelian monohybrid cross. Mendel has conducted his monohybrid crosses with all the 7 characters. Okay, but we are just studying only one, and if there is any problem question related to any other Mendelian monohybrid cross, you can make simple and cross like this. What you have studied in the monohybrid stem height cross. Similarly, you can make all other crosses using symbols. Okay. Anyway, there is one more law after Mendelian monohybrid cross. That is the second law, law of segregation. Second law is law of segregation. So after monohybrid cross, there is law of segregation. This law states that during gamete formation, the allelic pair segregate. You have already shown it, okay, in the cross. So that gametes receive only one of the 2 alleles. Mendel taught one of the 2 factors. Okay, so law of segregation states that it is related to gamete formation. So the statement should start with during gamete formation. Okay, law of segregation states that during gamete formation, the allelic pair, the factor pair segregates, separate, so that gametes receive only one of the members. But the 2 members, only one is born to one gamete. You know this theory behind it because there is chromosomes are getting half during meiosis. That will, or later we will mix it up. Okay, so this is the law of segregation. During gamete formation, the allelic pair segregates so that gamete receives only one of the 2 alleles. I think this clear. What is a monohybrid cross? A cross in which inheritance of single gene is studied. We are focusing on only one character inheritance at a time. For example, whatever the characters the plant had, Mendel focused only on the stem height, tall and dwarf. That only his account. Okay, so in a monohybrid cross, you have studied, okay, the first generation, raising up a first generation by crossing tall and dwarf plant. All of them were tall, okay, and genotype of the tall plants were capital T, small t in the F1. When the F1 were selfed, Mendel observed 75% of them tall, and 25% of them dwarf. And here he has observed 3 different genotypes, capital T, he is proposing 3 different genotypes, capital T, capital T, small t, and small t. So in the F2 generation, we have 2 ratios to study, phenotypic ratio and the expressed features ratio, 3 tall is to 1 dwarf, and the genotypic ratio, 1 capital T is to 2 capital T small t is to 1 small t. Homozygous, heterozygous, tall and dwarf ratio. Okay, so the phenotypic ratio is 3 is to 1, genotypic ratio is 1 is to 2 is to 1. Altogether, you have studied 3, there are 3 laws altogether. You have studied 2 laws of principles of inheritance. First law is law of dominance and the second law is law of segregation. I think all these concepts are clear for you. So anyway, we will do some more problems related to monohybrid crosses, that it will be very clear for you. And very important thing, all students should prepare note on this. Anyway, I will give a note, class note as a class work for you, and you can see that class note. And before that, you start your book with writing the name of the unit in the first page and all the chapters in that page, name of the chapters. The second page, you start with the chapter heading, Principles of Inheritance. The first page should be with the unit name, Genetics and Evolution, and the 3 chapters split up. Okay. The second page, you start with the chapter heading, chapter 1, Principles of Inheritance and Variation. And don't begin the classwork not there. You have to leave that 2 pages for writing the terminology of genetics. That also I will attach as a file, PDF file for you. See, listen, the first chapter, front page, you should write all the common terms used in genetics. Okay, I will give it. Terminology in genetics. Okay, and so many terms you have studied today. Dominant, recessive, gene, allele, homozygous, heterozygous, genotypic, ratio, phenotypic ratio, like all this terminology you should write first. Okay, so if you have any doubt, I will clear a doubt. Okay, thank you.